A comparison/contrast of both cell division processes. How many genetically identical daughter cells are created following cytokinesis? 2 genetically identical daughter cells are formed following it. How is the cell cycle, a complex set of stages, regulated via checkpoint? The cell cycle is controlled by a control system. By both internal and external signals, the cycle is regulated at certain checkpoints where stop and go signals regulate. How are homologous chromosomes oriented along the metaphase plate during mitotic metaphase and metaphase 1? In mitosis, chromosomes are duplicated to create 2 sister chromatids. The individual chromosomes are lined up at the metaphase plate during metaphase. In meiosis, chromosomes are duplicated and homologous chromosome pairs are held together by sister chromatid cohesion. The pairs line up at the metaphase plate. Homologs separate during anaphase 1 and the sister chromatids remain attached at the centromere, which separate later during anaphase 2. How do observable features within a cell change throughout the continuous mitotic process? Mitotic phase includes both mitosis and cytokinesis. In the stages, the cell grows, divides, separates, and forms 2 daughter cells. What occurs during each of the 3 stages of interphase? There are 3 interphase stages, G1, S, and G2. In G1, the cell begins to grow. In S, chromosomes are copied and DNA is synthesized. And in G2, growing continues and prepares the cell for division. 2 centrosomes are formed by duplication of a single centrosome. What is the relationship between homologous chromosomes and homologous chromatids in crossing over? 2 sister chromatids of each duplicated chromosome separate and move into 2 new nuclei, one forming at each end of the cell. Once the sister chromatids separate, they are no longer called sister chromatids but are considered individual chromosomes. This step essentially doubles the number of chromosomes in the cell. How does cancer result from disruptions in cell cycle control? Cancer cells do not need the normal signals that regulate the cell cycle. They may have an abnormality in the signaling pathway that conveys the growth factor signal to the cell cycle control system. Another possibility is an abnormal cell cycle control system. Since meiosis is a reduction division needed to create haploid gametes, which process restores the chromosomal number by creating a diploid zygote? The process that restores the diploid number of chromosomes is called fertilization. The genetic contribution of each parent is given in a single string of genetic code. Through fertilization, the genetic material joins to form the diploid number as a gamete. How do cyclins, cyclin-dependent kinases, and mitosis promoting factor relate to cell cycle checkpoints? Cell cycle checkpoints are control mechanisms that ensure the loyalty of cell division in eukaryotic cells. Cyclins and cyclin-dependent kinases are part of the G1 checkpoint, which transitions to the G2 checkpoint, which mitosis promoting factor is part of. How does mitosis pass a complete genome from the parent cell to the daughter cell, but meiosis ensures genetic reduction? At metaphase 1 of meiosis, chromosomes are positioned at the metaphase plate as pairs of homologs, rather than individual chromosomes like in metaphase of mitosis. Why does DNA need to replicate during interphase? DNA replication is a necessity for cell division. Interphase consists of G1, S, and G2 phases. DNA replication occurs in the S phase. It does so because the signals from the end of G1 phases activate proteins that begin to duplicate the DNA. How does crossing over events in meiosis create genetic variation? 2 divisions produce 4 non-identical daughter cells, each containing half the number of chromosomes of the parent cell. This provides an opportunity for new gene combinations to occur in the gametes.